Well, hello everybody. Now we'll go directly to the chapter number 6 in the textbook, and we will discuss selection for simply inherited traits. Actually, if we want to select for a simply inherited trait, you do need to know that we are dealing with a few number of genes, and this is much easier than the selection for the quantitative trait, which is controlled by so many genes. So let us start. We do need. I will give it to you as a briefly in this chapter. How can we select in general for simply inherited traits? As you see, for selection for simply inherited traits, we need only to know the following. Some information should be known before commencing in the selection for simply inherited traits. For example, how many loci are involved. And this is a little bit complex to know those things, but if you know the number of loci involved in a certain trait, it will solve a certain percent of the problem. Also, you do need to know how many alleles at each locus. Now we have the loci, how many loci involved in the inheritance of this trait. Then we will go further deeply to understand how many alleles that that can be found in this locus. In addition to that, you do need to know how those alleles are expressed. And this is very important. First of all, you do need to identify the loci or the locus that control the trait of interest. Then the expression of those alleles, in addition to the genotypes or probable genotype of the potential parents. This is very important to be known before start doing this thing. For example, if we go to the expression or the expressed alleles, I can give you an example related to the Khaleel gene. The Khaleel gene. The Khaleel gene inheritance is totally different from the Mendelian inheritance. It's called also the polar overdominance in it. Polar overdominance. This type of expression is totally complicated. This genes or those mutations can be found in the males and females, but none of them can be expressed, whether they are homozygous or heterozygous in the female. It will not express in the females. It will be expressed in the males that of heterozygous, not homozygous, but they can pass it to the next generation. Also, the expression of this mutation, the Khaleel mutation, for example, this is an idea. Why do we need the expression to be known? Also, for the Khaleel gene, you do need to understand that this gene can be also transmitted from the parent to the offspring. I mean the mutation. Okay, this mutation cannot be expressed in the females, whether it was homo or hetero. Again, also it will be expressed in the males who carry the mutation in the heterozygous form, not homozygous. Okay, and also those males those males that inherited this mutation from their dams or mothers, they will not express it unless they take it from their parents, from the parental or from their sires. This is an idea how the expression should be involved. Why do we need this thing? Because if you understand the mode of inheritance, the alleles found or expressed in the individuals, and if you know the genotypes of the parents, I mean the dams and the sires, you can start the selection process. Because, as you see from the Khaleel expression mode, the polar overdominance is very complicated, but there are a lot of expressions or genes or mutations that also with much easier expression than the other. For example, another example here, I can told you about it. It is the FIC-B gene, that controlled the prolificacy of the sheep, for example. Now, this gene or this mutation can be found in a certain chromosome, and also it can be inherited from parents, from the dads, and also from the sires, from the mother and the father, and its expression also follow what we call the additive expression. It is additively expressed. So how it comes? Now, 3 forms of those genotypes can be found in the individual. One of them is the wild type, which is doesn't carry the mutation, and also it will be B+, which is heterozygous, just only one allele from this mutation, and also the homozygous, actually, the homozygous of those genotypes. So it will carry 2 copies of the alleles, B. So we have B+, B+, and also BB. So as I told you previously, that it is of additively inherited. So by increasing the number of B's, the expression of this gene will be much more than the others. So if you have, for example, ++, it means that the mutation is not found in that individual. It will give a normal range of prolificacy. But if you have B+, for example, that holds just only 1 copy of the gene, the prolificacy will be increased in a certain amount, a certain level. It means the allele will produce much more twinning rate, much more individual, much more newborns per lambing, okay, than the ++. But if you go for BB, for example, as I told you, additively, by increasing the number of the copies of the B's, okay, the prolificacy will be increased much more than the 2 genotypes. For example, if the B+ produces 1.7 as a prolificacy, or lambs per lambing, in some certain papers I see that it will produce 2.3 lambs per lambing as an average. Okay, and from our experience, it produces such level of prolificacy. So you can go back, if you want to some details about this mutation, you can go back to my papers that can be found if you search in Google. You can find the cleavage mutation paper and also the prolificacy or the FIC-B mutation. So why do we need such information? In this case, we do need to select those animals that carries the favorable alleles or mutation. But in some cases, and in all cases, the mutations are of bad influence on the animals or on human in general. So in some cases, we do need to select those animals to keep them to be parents for the next generation if they hold a certain favorable mutation. But in most cases, the mutation has a bad effect on the animal. So we do need, in some cases, we do need to discard or to cull them from the flock in order to prevent the animals from genetic defects or any other diseases that can be caused by mutation. So breeding for simply inherited trait involves methods to ensure the genotypes of the individual. So we do need to understand the 4 points, or to know the 4 points that mentioned here: how many loci, how many alleles, how those alleles are expressed, and the genotypes of probable genotype of the potential parents. Now, if you are dealing with certain mutation that cause, for example, a certain change, a certain obvious change in the phenotype, you can simply select for it. For example, we have, as you see from those pictures, that we have a certain trait. We take here the coat color in the Shetland sheepdogs. As you see, the homo dominant or homo recessive, those genotypes produce a distinctive color for those dogs. But the heterozygous, as you see from here, this one to the right of you, okay, it will produce a certain different color from the homo dominant or homo recessive. So it's very important or very easy to select for those animals, because this mutation produce a very obvious phenotype. This phenotype is clearly distinctive. You can identify those heterozygous from homo recessive or homo dominant. So it's very easy to select or to cull the preferred individuals. And as you see from here, if we use what we call the Punnett Square, if we mate two individuals of heterozygous, okay, two heterozygous individual, M capital with M small and M capital with M small, the productivity or the new offsprings will be as you see here, M capital, which will be of distinctive color. M capital, M small, it is a hetero, also it is different color from homo recessive or homo dominant, and also as you see M small, okay, it is also of different phenotype. So it's very important and very easy, because there is just only one locus involved, and also the phenotype is totally distinctive, مميز, distinctive from the homo, the hetero. You can identify the hetero or the homo recessive or homo dominant from the appearance of those animals, from their phenotype or the color code.